glucocorticoid responses, personality, and ability of animals to cope up with changes in their environment. I request John to please deliver his lecture. Good morning. Good evening in New Zealand. Or is it about to be good night? I don't know.
Good morning, and thank you for the opportunity to contribute to the conference. It is the afternoon here in New Zealand.
Good morning. Michael.
I will share my screen to begin my talk.
Okay, go ahead.
Good morning, Michael. So can people see my screen?
Yes, it is visible. You have a total time of 15 minutes and 5 minutes for interaction and question answer.
Okay. Thank you. So the title of my talk, Individual Variation, Glucocorticoid Responses, and the Ability of Animals to Cope with Changes in Their Environment. So I'm interested in responses of whole animals to changes. And we have the overarching question, how do animals cope with changing environments? And then in particular, how is the endocrine system, especially the HPA axis, involved in the responses of animals to changes in their environment? So we can understand the role of the HPA axis and then go on to ask, how can we use endocrine responses to predict the ability of animals to cope with changing environments? This is a scheme that illustrates the HPA axis, which we're familiar with. So stimuli that we can call physical stressors or emotional stressors result in activation of the hypothalamus, and via CRF and the pituitary gland and ACTH, then there is activation of the adrenal or the intrarenal gland and the release of the glucocorticoid hormones, either corticosterone or cortisol. And these hormones have metabolic actions, they influence behavior, and they have other actions as well. So I'll turn now to consider individual variation in responses of animals to changes in the environment. And we're looking at the HPA axis and responses of glucocorticoids. And in this example, for Japanese quail held in a captive situation in Tokyo in Japan, we have on the x-axis time 0, 15, and 30 minutes, on the y-axis corticosterone concentrations, and the graph shows the mean concentrations in approximately 60 quail that were handled for 30 minutes. So we can see that the mean concentrations increased from 0 to 15 and from 15 to 30 minutes. However, if we have a look at all the individual responses, we see a different picture. We can see that some birds had very high responses, whereas other birds had virtually no response. And so the original statement that corticosterone increases in quail handled for 30 minutes, in fact, only applies to the mean values. In reality, we can say corticosterone increases in some quail handled for 30 minutes, whereas there is no change in corticosterone in other birds. So these birds have all had exactly the same stimulus from the environment, but there is this great range of responses. And we see this in every species of vertebrate that we have considered for glucocorticoid responses. I turn now to a very different species of animal. This is the Gnrh, the little penguin down here in New Zealand. On the left, the mean corticosterone responses of 100 penguins. And you can see that the mean response showed a very clear increase up until 30 minutes, and then little or no significant change from 30 to 60 minutes. For the individual responses, as we saw previously for the Japanese quail, we have a whole range of individual responses. At 60 minutes, some birds had corticosterone almost 300 nanograms per mil, whereas other birds' corticosterone was only perhaps 10 nanograms per mil. So these are wild birds all faced with the same stimulus, and they have this range of responses. We were able to sample some of these birds out in the wild 1 year after the first sampling occasion. And we see in this graph responses of 4 individual birds sampled at an interval of 1 year. And this bird had almost the same response a year later. Similarly, the bird on the bottom left, bird on the bottom right, this bird. So these differences in responses are individual characteristics. They are consistent. They are a feature of the individual animal. The third example of individual responses is from another penguin, this time the Adelie penguin down in Antarctica. And here we have a range of responses of the same birds sampled on the first occasion, about 5 days later, the second occasion, and then another 5 days later on the third occasion. If we have a look at the bird shown in red, it had virtually no response on the first occasion and the lowest response of any bird. Again, on the second occasion, the lowest response. On the third occasion, almost the lowest response. Another example of these consistent individual differences in a glucocorticoid response to a threatening stimulus. So we have these consistent physiological responses to a stimulus from the environment, and these physiological responses are linked with behavioral responses of animals. And in a moment, we'll talk about how we call the linking of the behavioral and physiological responses personality. Just before that, another slide from the Adelie penguin showing for 2 individual birds, number 21 consistently had a low response in comparison with bird number 27. Now we get to this term personality. So we can define personalities as consistent individual behavioral and physiological responses of birds and other animals to changes in their immediate environment. We've seen these consistent physiological responses in the form of the glucocorticoid responses. The physiological and the behavioral responses are linked and into what we can call for animals' personalities. And we can classify animals into 2 personality styles, the so-called proactive animals, which are the active and bold animals, in comparison with the so-called reactive animals. These are the passive and shy animals. This is a scheme that I use to think about the linkage between the physiological and the behavioral response. So in response to a stimulus from the environment, there is the process of perception, and then the individual characteristics of the animal, which we call personality on a spectrum from proactive to reactive. These individual characteristics determine both the behavior response and the glucocorticoid response. So the endocrine response does not cause the behavior response or vice versa. The endocrine and the behavior responses are both a consequence of the individual characteristics of the animal. This is a table showing some of the characteristics of proactive in comparison with reactive animals, some of the behavioral and characteristics, and then the glucocorticoid response. The proactive animals, these are the ones that faced with a threat, they will either meet the threat in a fight situation or move away. They tend to be aggressive and bold. When it comes to exploration, they're fast but relatively superficial to explore. But then in terms of flexibility, they're not well able to cope with a new situation. They're relatively rigid and routine-like. They show less fearfulness when experiencing a threat and have the low glucocorticoid response. So we saw that range of glucocorticoid responses the low glucocorticoid response animals will be the proactive ones. In comparison, reactive animals, in terms of behavior, faced with a threat, they will freeze or hide. They're relatively non-aggressive and cautious in social situations, slow to explore but thorough. And then in terms of flexibility, these animals are relatively flexible, so they are able to adjust to changes in circumstances by changing their behavior. And then we come down through fearfulness to glucocorticoid responses. So these are the high glucocorticoid response, what we might call the high stress animals. These animals effectively are very sensitive to changes in their environment and well able to change their behavior. The animals that are the low stress animals are less sensitive to their environment and less able to change their behavior. Introduce another term for animals. This is for wild animals, and we talk about fitness, the production of offspring that survive to breed. So a genotype's fitness includes its ability to survive, to find a mate, produce offspring, and ultimately leave its genes in the next generation. So an animal that has high fitness contributes to the next generation. Another one of my schemes, this time thinking about fitness. So here we have the stimulus from the environment, perception by the animal, individual characteristics, which we call personality, determine the behavior response and determine the glucocorticoid response. And together, these will influence the fitness, the breeding success, the survival of the animal. So glucocorticoids and behavior both influence fitness. It's been found that the animals with low glucocorticoid responses and proactive personalities have higher fitness in constant conditions. So in relatively constant conditions, these animals do well. But the high glucocorticoid response, high stress, reactive personality animals have a higher fitness in changing conditions. So if conditions are changing, the high stress sensitive animals may contribute more to the next generation. So in terms of glucocorticoid responses and personality and fitness, we see that the relationships between these, between glucocorticoids, personality and fitness, depend on the prevailing environmental conditions. And hence, for the wild animals, there's no optimum or best glucocorticoid response for all conditions. So we see this huge range of glucocorticoid responses, a range of personalities. In some situations, the low stress animals will do best. In other situations, the high stress animals will do best. The last term that I'll introduce is phenotypic plasticity. So this is a term that relates to the capacity of an animal to change a physiological or behavioral variable in response to a change in environmental conditions. So plasticity is the ability to change a behavior, to change a physiological variable in response to a change in the environment. And we can look at plasticity in relation to glucocorticoid responses. And I've made up a hypothetical example for cortisol responses of 3 species of fish. So on this graph, the y-axis will be the size of a cortisol response. The x-axis I've represented 3 water temperatures, 20 degrees, 23 degrees, 26 degrees. So down the bottom, the species of animal A, the cortisol response at 20 degrees is the same at 23 and the same at 26. But if we have a look at the animal up here, this animal called B has a cortisol response at 20 degrees, a higher response at 23, and a much higher response at 26. So we can look at the cortisol response of an animal in different situations to see if there is plasticity in the response. Does the response change and determine in relation to the conditions? And the slope of this line is what we can call a reaction norm. And we can look at reaction norms in relation to environmental stimuli as a measure of plasticity, as the measure of the ability of the animal to change. So for the glucocorticoid reaction norms in this example in fish, we've just said the slope of a glucocorticoid reaction norm indicates the plasticity of the response. Individuals and species will vary in the plasticity of their responses to changes in water temperature. It may be that an increased response is good for the fish. It may be that an increased response at a higher temperature is bad for the fish. Nonetheless, the reaction norms for different populations and species may be useful for predicting how fish will be able to cope with increases in water temperature associated with climate change. So I'm presenting this as an example of how looking at the glucocorticoid responses may help us to predict how animals will cope with changing environments. So the conclusions. We've noted that, of course, the HPA and the HPI axes and glucocorticoid responses are central to the responses of animals to changes in their environment. We know, of course, that the natural environment is changing due to climate change, with greater frequency of weather extremes, increasing air and water temperatures. And so finally, we can have suggested that glucocorticoid reaction norms may be useful for predicting the ability of animals to adjust to changes in the environment due to climate change. Thank you for the opportunity to speak. I'll stop sharing my screen now.
Thank you. Thank you very much, Professor Cochrane, for a wonderful talk. It's open for discussion, and it's ended in time, well in time, so we can have more than a couple of questions. Dr. Asuman.
Good morning, sir.
Good morning.
Yeah, actually, as you were saying, there is a central response for the behavioral purpose for the glucocorticoids. But as we know that the peripheral and the central, both organ are very much, linked in the response, especially in case of behavior. So, like if you see in case of penguin, also in your last presentations, we have seen that they have a steep level of cortisol, but when they are residing in the fridge, when they are becoming freeze, that response is reducing. So in that case, how do you say that it is only a central response? That's why I was just wondering, that is it only the central response?
So is the question, is the HPA axis response always driven from the central nervous system?
Yeah, but it is a coordinating response. The organ which is producing the glucocorticoid with the signal of the hypothalamus or the brain, and then it has a signaling which is coming through various pathways. But it is a coordinated response from the glucocorticoid production center and the signal received by the brain that, okay, the outer environment is like this and that, and you have to respond accordingly. So in that case, is it only the central thing, or the organ which is secreting the glucocorticoid that is also having a role to give this axis functioning?
So in some situations, people have conducted experimental tests to ask about the sensitivity of the pituitary gland to CRF, the sensitivity of the adrenal gland to ACTH. And whilst this has only been done in a handful of species, my sense is that the primary source of the variation remains the hypothalamus at the top of the axis.
Thank you, sir. Thank you.
Professor Michael.
Thank you, John, for the useful presentation. And I could see a consistency in your finding for different animals. I have one clarification. you are concentrating on the HPA axis. There are other stress hormones like leptin, thyroid, epinephrine, norepinephrine. Is there any data on those hormones in these animals?
So is the question about data on individual variation in other endocrine axes?
Stress hormone, other stress hormones.
So there is surprisingly little data actually on variation in individual variation in other endocrine axes. People very often they consider variation to be a nuisance in relation to the question that they are asking. And so it takes a change in perspective to ask particularly about variation and the importance of variation. Well, I'm working with a frontiers research topic at the moment about individual variation, and somebody has been looking at variation in the IGF axis, insulin-like growth factor 1. And so this is a relatively rare example of where people are finding consistent variation in other endocrine axes.
Thank you.
A quick question from my co-chair. Thanks, Professor, for the wonderful work. The question that is in my mind is regarding the role of nervous system organization at the phyletic level, fish to mammal. In your case, you have done both fish and birds. A higher organizational central nervous system, in other words, higher phyletic species, the response will not be the same as fish. The reason is the higher level of organization will allow this individual to assess the world around it in a much more complex way. So, like in this case, the solitary species and the colonial species, the response would not be the same. Any light you can throw on this? Based on what we know. The solitary species and the colonial species. And the lower organism and the higher organism, the way the stress produces the response in terms of cortisol would not be same. As you have already shown, you classify it into two categories. The low response species and the high response species. Similarly, I expect differences in the colonial species and the solitary species. The prey species and the predator species. The lower organism and the higher organism. Can you educate us on this area?
So the audio quality is a little hard to follow. Sorry, what was the question about responses of fish, colonial fish versus other fish? I'm sorry, it was difficult to catch through the microphone the sound of the question.
I think we can just okay. Yeah, we don't have time.
Okay. Okay, John, thank you very much for participating in this event. Your presence makes a lot of difference. And the senior members will be aware of the fact that John Patra is responsible for the SRBC getting affiliated to Asia Oceania Society for Compared Who is one of the founders of that AOAC, and many of our colleagues in the service, like Damu Kumar Mehta, Samir Bhattacharya, or so like that, we were having wonderful opportunity to interact with them in Asia Oceania. And visibility of SRBC also increased internationally. So it was the first international association to recognize that. Thank you, John, for joining us.
Thank you for the opportunity.
Thank you very much, Professor John Patra, for fantastic talk. We move on to now the invited talk number 25, IS 25, by Dr. Yuvraj Sambandam. He will be joining us from US. We have preponed this talk due to the suitability of the time zone. Otherwise, it will get very late in US. Dr. Yuvraj Sambandam is an assistant professor of research at the Department of Surgery, Comprehensive Transplant Center, Northwestern University, Frinberg School of Medicine, Chicago. He is going to talk today on kidney transplantation, diagnose rejection with the donor reactive T cell clones. Dr. Yuvraj is there online. And
Yes.
we can see your screen clearly, and you have 15 minutes for the talk and 5 minutes for the discussion. Over to you.
Okay. Are you able to listen? Am I audible?
Yes, very well.
Okay. Thank you so much for the nice introduction. And first of all, I would like to thank the organizers, especially Professor Malini, for inviting me and giving me the opportunity to present my research here. It is always a pleasure to deliver a talk to SRBC with my friends and professors are there. So today I'm going to talk about how the we are actually diagnosing the rejection with the donor reactive T cells in kidney transplantation. We are devising the methodology to diagnose the rejection. So that's what I'm going to talk about today. The solid organ transplantation is one of the most complex area of modern medicine. It has been a life-saving therapy for many patients who have end-stage kidney diseases. That is called ESRD. The patients who have end-stage kidney disease, stage 3 to 4, are recommended to have a new kidney. In Northwestern medicine, the transplant program started at 1964. So far, more than 5,000 kidney transplantation have been performed. And hence, the Northwestern medicine is one of the largest living donor kidney transplantation programs in the state as well as the country. There are many challenges for the transplantation, and one of the important challenges to keep the transplanted organ for the long time. In order to keep the long-term survival, we need to have effective tools to diagnose the cause of allograft dysfunction. If we have a correct diagnosis, it is a critical point where the doctor identify the problem and then prescribe the immunosuppressive regimen to keep the transplantation safe. And this graph shows what's the importance of the subclinical diagnostic tool so that we can identify the rejection and then prescribe the treatment. Here, the blue line represents the function of the transplanted kidney. When the acute rejection happens, the function of the kidney is dropped down. So acute rejection can be identified by looking at the function of the kidney, that is estimated by glomerular filtration rate. But if subclinical rejection happens, there is no tools to identify subclinical rejection. This is very important. If there is no tool, so if there is no identification of subclinical rejection, that leads to the acute rejection and then followed by the failure, graft failure. So we are working on how to identify the subclinical rejection here to keep the transplantation in a safe way. There are major risk factors for graft failure. One of the important factors is donor reactive T cells, which appears before the subclinical rejection. That is, and the donor reactive T cells appear before the subclinical rejection so that we can identify the upcoming rejection. However, there is no tool to identify the donor reactive T cells within the allograft. So we have previously developed this technology with Adaptive Biotechnology, that is a company, and we have a patent together with Adaptive Biotechnology company. What they do is, like, they sequence the T cell receptor beta gene, that is immunoseq analysis. By doing the sequence, they identify the donor reactive T cells. So what we are doing is, by looking at the donor reactive T cell in rejecting patient and stable patient, whether we can identify the upcoming rejection. So the immune diversity is generated by the recombination of many three genes, like variable gene, donor gene, and joining gene. The recombination of these three genes, which makes the TCR receptor, which is on the T cells, that is the unique sequence. So because of this is the unique sequence, we used as this sequence as the barcode to identify the specific clone, that the specific donor reactive T cells. Then we can track the specific clone throughout the period. So when you take what is the diversity of this human, the donor reactive T cells? It's very huge, 10 to the power of 13 different clones. Therefore, we need a specific, precise method to analyze the donor reactive T cells. That's where we were applying immune repertoire analysis, that is called immuneseq. With this background, our aim of the study is to diagnose the kidney transplantation rejection by monitoring donor reactive T cell clones in post-transplantation, biopsy, and non-invasively in blood and urine samples. Further, whether we can diagnose subclinical rejection or predict progression from subclinical to clinical rejection. Coming to the experimental design, we have 4 parts of experimental design. The first one is pre-transplant, and second one is post-transplant, and the tracking of the clone, finally diagnose. In the pre-transplant condition, what we have done is we isolate the PBMC from recipient and donor PBMC, and we have done MLR, which is a mixed lymphocyte reaction before the transplantation, and then allow the cells for culture. We culture the cells for 7 days. And after that, we have identified the proliferation of donor reactive T cells. From there, we isolate CD4 and CD8 cells, which is specifically donor reactive recipient T cells, which is called DRTC. And these cells we were subjected to immuneseq analysis, that is nothing but TCR beta sequencing. The second part is post-transplantation. After the transplantation, in different time point, we collect blood, urine, and kidney biopsy. And then these samples subjected to immunoseq analysis, that is the same analysis what we have done in pre-transplant condition. The third one is we are tracking these clones like pre-transplant donor reactive T cells in post-transplant samples. By tracking these donor reactive T cells, whether we can diagnose or predict an upcoming rejection. How we generate the donor reactive T cell and using the pre-transplant MLR. Here the lymphocyte. From the lymphocyte, we have gated the responder cells, that is the proliferating cells. From there, we isolate CD4 and CD8 using the flow cytometric analysis and use the cells for the further experiments. Coming to the result here, this graph shows the abundant expression of DRTCs in different time points, that is pre-transplant condition, 3 months at rejection. We have done many patients, but I am showing here only 2 patients, patient 4 and patient 20, DRTC, CD4 DRTCs and CD8 DRTCs. You can see here pre-transplant condition, there is no DRTCs present, but at 3 months, there are a lot of DRTCs are appeared before the rejection happens. The rejection episode happened at 4 months in patient 4, at 6 months in patient 20, but we are able to see the donor reactive T cells at 3 months itself, which is indicating that the donor reactive T cells, if you have the donor reactive T cells, then it is going to be a rejection episode. We are going to see the rejection episode in the same patient. So this is in biopsy. We are actually looking at whether we can see the same result in PBMC also, because biopsy is the invasive method. So we wanted to see whether we can see the same result in non-invasive method, something like isolating PBMC or urine. So next we see the same result in PBMC. In the pre-transplant condition itself, CD4 DRTCs and CD8 DRTCs are more. And the DRTCs are also present at 3 months and 6 months. And eventually, whoever the patient showing the DRTCs, more DRTCs, they end up with rejecting the allograft. Similarly, we have done the same experiment with urine, because urine is very simple. If you able to see the same result in urine, then it is very easy to identify the rejection episode before the rejection happens. So we have done 2 weeks, we have taken the 2 weeks urine because we don't see any urine before the transplantation. So we take the 2 weeks urine as a base. At 3 months, 6 months, you can see at 3 months, there are number of DRTCs identified. Even though at 6 months, the number of DRTCs is less, but later at 6 months itself, the patient shows the rejection episodes. So at 3 months, in urine itself, it is showing that DRTCs is more, so it is going to be a rejecting episode. This graph shows, this is the actually Venn diagram showing that how we are tracking the donor reactive T cell in different time points. As you can see here, the donor reactive T cells are in pre-transplantation condition, which is also, we are also tracking those clones in 3 months, 6 months, and 12 months of post-transplantation condition between stable and rejection. You can see the clear demarc between stable and rejection. You can see at 3 months, a very low number of DRTCs, but at the rejection patient, you can see the more number of DRTCs. When compared to rejection and stable, we'll see the difference, clear difference between the appearance of DRTCs. Also, urine and kidney, you can see the clear expanded clones, more clones at 3 months in kidney as well as urine as well as PBMC. So what we have derived from this analysis is, like, we are able to track the clone in all throughout the period, as well as we can see the clear difference between stable and rejecting patient. This graph shows the cumulative result of all stable as well as a rejecting patient. We have done 18 stable patient and rejecting patient. Here you can see at 3 months kidney biopsy. This is highly invasive method. People don't like it, and that's what we are going to divert, or alternatively whether we can analyze the same result in PBMC and urine. That's why we are comparing the kidney biopsy, urine and PBMC. This cumulative result also showing the same pattern that when compared to stable, the rejecting patient shows high level of DRTCs. When come to the urine, the same result, like we are able to see the same result in urine as well as PBMC. This is actually at CD4 DRTC. We are also looking at the CD8 DRTCs, whether it is the same case in DRTC, CD8 DRTCs. Here you can see in the kidney biopsy at 3 months, the rejecting patient shows high level of DRTC when compared to the stable patient. In the same case in urine as well as PBMC. So I stop my presentation here by concluding. This is actually ongoing project. We have only taken 30 patients who are completed 1 year period. So in this interim result, what we have suggesting is MLR DRTCs, that is pre-transplant DRTCs, can be utilized as a diagnostic tool to identify the ongoing rejection as well as even predict an upcoming rejection, that this can be achieved non-invasively in blood and urine. However, a definitive conclusion can be drawn only after completion of the whole study. I would like to acknowledge my directors. Dr. Leventhal is the surgeon who done all the kidney transplantation surgery, and he is an interim chief of Comprehensive Transplant Center, Northwestern Medicine. And Dr. Matthew is the director of the lab, and people who were helped in this project. And also like to thank DOD for the grant support. Finally, I would like to thank all the participants and donors to support this study. And thank you for listening. Thank you so much.
Thank you, Dr. Yuvraj, for a wonderful presentation and being perfectly on time. It's open for discussion now. We can have a couple of questions. Well, I think there are no questions or suggestions from the audience. So in that case, I once again thank you for a very nice presentation. Can we have a round of applause, please?
Thank you. Thank you. Thank you so much for having me.
Now we move on to the invited talk IS 22 of Dr. Patra Roy. Dr. Patra Roy is a professor at the Molecular Endocrinology Lab, Department of Biosciences and Bioengineering, Indian Institute of Science, IIT Roorkee. He's a PhD in Life Sciences from Visva University, and his area of research is animal biotechnology, molecular endocrinology, mechanism of hormone action. He's also an FRE of the Society for Reproductive Biology and Comparative Endocrinology. He's going to today talk on camperferol, a major phytochemical that creates that's critical role in protecting pancreatic beta cells and preventing obesity-related diabetes. Dr. Patra Roy, we can see your presentation now clearly, and your time is 15 minutes and 5 minutes for the discussion. Over to you.
Okay. Thank you very much, all of you. So it's always a pleasure to present our work in this SRBC. I am sorry I could not present myself physically. I wanted to be there in the meeting personally, but somehow due to some restrictions in travel from the institute, I could not travel. So I'm sorry about it. So thank you very much for giving this opportunity. So I will talk about some of our ongoing work in our laboratory, because basically we work on phytochemicals and their role in alleviating different diseases. Today I am talking about the effect of an phytochemical called camperol on the function of the pancreatic beta cells. Okay, so So what happens? Why you are so much concerned about the pancreatic beta cell functions? So you see, this is adipocytes, and during the obesity condition, the adipocytes accumulate large amount of fats, and then there is an accumulation of macrophages, which starts to do the destructive activities of oxidative stress, DNA damage, telomerase dysfunction, and to protect itself, function of these fat cells. So there is upregulation of p53, which actually in turn causes the pro-inflammatory cytokines to go up, insulin resistance, and ultimately leading to diabetic condition. So diabetes and obesity are, that is why, highly closely linked subjects, and they are occur simultaneously. So we are, with this question in our mind, we wanted to address this issue in a better way. And so we wanted to see if there is any natural products which can address the issue of obesity as well as diabetes. So because our goal is to make the foods as a drug rather than consuming any drugs as such. So when we searched for the literature, we found that there are several flavonoids, and out of these, flavonols has been reported to be a very critical and important class of molecules which can induce the anti-diabetic properties. And but
What's your audio is off?
I think there is loss of network from his end. Dr. Patra Roy, you're not audible. Can we just make a phone call and inform him that he is not audible?
No, Master. You're off. You're off. What's the problem there? I think the problem is you.
He can log out and then again log out.
You can log out and try.
Your voice is not audible, Dr. Patra.
yeah.
Please share your presentation.
So yeah. So is it okay now?
Yeah.
So I'm so sorry about this interruption. So I think you can see the slide which I was showing.
Yes, yes, you can see.
So okay, so sorry. So we picked up the camphorol as an important molecule because it is sold commercially for various applications. That is why we selected this camphorol, and this is how this happens. So in the diabetic, in the normal conditions, there is a blood glucose goes up, and then blood glucose induces the pancreatic beta cells to release insulin, and then insulin then acts on the other targets to liver, muscles, and all, and then ultimately the glucose gets absorbed. But in normal conditions, otherwise, when the obese condition comes, free fatty acids is upregulated, and this actually leads to the production of resistance of insulin resistance in the muscles and liver tissues. Also, it causes the pancreatic beta cells functional failures. So this is the question we wanted to address, and for that we utilized our various approaches in our study. So let me move first. So our approach was to see the regulation of autophagy process in the pancreatic beta cells. So what is basically autophagy process? So autophagy is a self-destructive process in which the cell actually rejuvenates itself and then it tries to protect itself when there is any damage in the cell or in the organelles, etc. So it takes place in 3 different ways: macroautophagy, chaperone-mediated, and microautophagy. So in beta cells, this is the process which happens in the beta cells. So I will not go details of this one because this is the process of different types of autophagy process. So I will talk about the macroautophagy, where this the organelles to be destroyed are covered up by the phagophores, and then this phagophore fuses with the lysosomes, and once the fusion occurs, so then the cellular organelles or destruct which is to be destroyed, they get destroyed. So this is the pathway we have been following in the pancreatic beta cells. So what happens in the pancreatic beta cells? Actually, in normal conditions, when there is any damage in the pancreatic beta cells, so autophagic process starts in. And if it happens, so then then the pancreatic beta cells regenerates and then no diabetic condition occurs. But if the autophagy is inhibited, so then the pancreatic beta cells dies, proliferation is less, death is more, and the diabetic condition starts. So this is what we wanted to address, how the autophagic process can be regulated by using the camphorol. And free fatty acid is a process which actually inhibits the autophagy activity in a pancreatic cells. That was, we want to show a link how camphorol was elevating or this entire process. So our laboratory, so this is the work I am presenting by Dr. Ritu Bharshne, who finished her PhD in my lab, and that is what I am trying to explain some of the work that we are doing currently. So first wanted to see what is the role of camphorol in pancreatic beta cells functions. So we did both in vitro and in vivo approach. So first we address the question using in vitro approach. So this is the RIN5 cells we got from ATCC, and we tried to grow them in our conditions. And first question you want to address, what camphorol does? Does it protect the cell or it kills the cell? So when you treated the cells with RIN5 cells with camphorol, we could see that it was allowing it to proliferate. So proliferation was taking place. So that means it was not toxic to the cells. And we tested at different concentrations. It was rather initiating the proliferation of the cells. So we are sure that this is the molecule which can be a good target or good molecule to be used for future studies. So now we wanted to check, does it prevent the fatty accumulation in the liver pancreatic tissues? So we use palmitic acid as a molecule, which is basically a fat molecule and causes destructive effect in the cell. And as you can see here, when you treat the RIN5 cells with palmitic acid, there is deposition of fats in the cell. But once you treat the cells with the camphorol, you see all the fat both disappears. There is no fats, and even the palmitic acid induced fat was disappearing in response to camphorol. So that means it was inhibiting the fat deposition in the pancreatic cells, and this is very critical part of it. So we estimated the triglyceride levels, we estimated the oil water staining, and body P is basically a fat deposition marker. And as you can see here, the body P staining, which was seen in response to palmitic acid, was absolutely removed off in response to camphorol. So camphorol was preventing the fat deposition in the pancreatic cell, and this fat deposition in the pancreatic cell was very, very deleterious for the cells. So naturally, our question was how it was doing it. So then what we wanted to see that if the fat deposition takes place in the pancreatic cells, so this is the palmitic acid, you can see the cell dies. That means more fat in the pancreatic cell, more cell death. And as you treat them with the cells with the camphorol, then it protects the cell from death. So this was seen by MTT assay. And as you can see, palmitic acid treatment of the cell caused DNA ladder. So apoptosis was taking place. But if you treat them with the camphorol, that was gone. And also by this staining with the DAPI staining, you could see the nuclear condensation in response to palmitic acid, but camphorol could protect that one. So camphorol was protecting the fat deposition and at the same time, the cell death. Now the next obvious question that we generally do for any routine practice. So we checked that does camphorol protect the cells from apoptosis? So you can see here, caspase activity, which was going up in response to palmitic acid, was significantly inhibited in response to the increasing concentration of camphorol. And all the markers were downregulated, and at the same time the caspase 3 protein level, which is a palmitic acid, you can see upregulated. That means cells were dying, but with camphorol that cell proliferation was that means cell death was reduced. So it was reducing the cell death, rather it was enhancing the cell to proliferate. So rejuvenation of the pancreatic cell. So temporal must be used as an autophagy inducer, so which was doing the same function as this camphorol was doing. So now camphorol induces autophagy in palmitic acid induced stem cells. So that was our question was. So as you can see here, when we talk about autophagic process, it involves different stages: induction, then phagosome assembly process, then sequestration, and then fusion with the lysosomes. And we use two different markers, MDC, which is initial phase of autophagic marker, and we use lysotracker, which is the late stage of autophagic marker. So you have to see where the camphorol is acting. It was acting at both the stages or at different. In addition to that, we used different markers in this process. So this is the markers we used for here: AMPK, mTORs, ATG7. These are all different proteins which are responsible for autophagic processes. And we use MDC as an lysotracker as an early and late stage markers. So this is how we did the study, and we could see the palmitic acid's role and the role of camphorol in that entire process. So as you can see here, if you treat the cells with a palmitic acid inhibits autophagic process. So it inhibits early stage autophagy as well as the late stage of autophagy process. But in presence of, if you see the presence of camphorol, you can see it induces the initial phase autophagy process as well as late. And if we merge them, so it was showing that it was inducing autophagy, same as temsirolimus, which is positive autophagy inducers. But if you use palmitic acid, both the autophagy process was inhibited. So palmitic acid was inhibiting the autophagic process, and camphorol was inducing the autophagic process. So now we needed some marker, how to identify what is happening in the cell. So we recruited different protein markers. So these are the different markers which we use for autophagic process. One is LC3D protein, which is upregulated, and there's another protein called P62 protein, which is generally downregulated when the autophagic process is completed. So these are the two markers which we used in our study for experiments. So one important experiment that we did was this LC3 puncture formation. What is that actually? So when the autophagic process takes place, there is a complex formation between LC3 to P62, which is basically responsible for ubiquitination. So once this complex is produced in the cell on the autophagosomes, and that destroys the cellular organelles proteins, which is to be destroyed by ubiquitination process. That is what we have been looking for. So in a cell, you can distinctly see this puncture formation. More puncture formation means the cell is undergoing autophagic process. And if it is not there, cell is not going autophagic process. So we did a very simple but very elegant experiment in that way. What we did here, we transfected the cells with EGFA LC3 protein. So in normal condition, EGFA LC3 is distributed in the cytoplasm. Now, if you induce the autophagy in these cells, so what will happen? So autophagosome starts to form. This is the autophagosome starts to form the cell. And then in response to ATG7 other factors which induce autophagic processes, we could see the formation of the puncta on the autophagosome surfaces. So, so now you see all the green color, which is LC32, which was there, they now accumulates on the surface of the autophagosomes. And then subsequently they will be degraded out. So that's the same thing you can see here. When the BSA treated cell, no autophagic process is there, all the cell is green. But as soon as you treat them with the camphorol, then autophagosome starts to accumulate. So this is the basic formation of puncta. But if you treat the cells with palmitic acid and temsirolimus, which induces, you can see the same responses. But with palmitic acid, it's almost same as that of the BSA when the autophagy is not taking place. But if you treat with camphorol, again the autophagy process comes back, because you can see the puncta formation as good as this only camphorol, but it cannot rescue completely, but it is much better than what you see only with palmitic acid. So that means palmitic acid induced autophagy formation was rescued by the camphorol within the cell, which we could show by using this fluorescence pattern in the cells. So naturally, the question comes: what was it really mimicking the same what I was talking about? As you all know, I just not told you, LC3 protein and P62 are two independent markers of autophagic process. So once palmitic acid is treated, you see the LC3 protein, which is less, P62 protein is high. But if you treat them with temsirolimus positive autophagy marker, it's just the reverse. P62 goes down and LC3 level goes up. Same thing was shown in our camphorol also. LC32 was high and P62 was low, which is a typical indication of an autophagic process. This was dose dependent and with time also. You can see as you treat the cells with camphorol, the level of P62 drops down, LC3 level goes up. So that was very clearly showing that camphorol was inducing autophagic process in the cell, and that's how it was rescuing the cells from the for the degradation or these proliferation. So now, how do we establish that? Is it really true what we are showing? Autophagy inhibitors. If I use autophagy inhibitors, does this process get inhibited? So we use different inhibitors of different stages. Morphonin, AT7, siRNA, chloroquine. These are different stage inhibitors in the cells, and we could say, I can just summarize this part very fast, and I can show you that once we block these processes, so as you can see here, once we block, this is palmitic acid. As usual, there is no autophagy process. If you treat them with the camphorol, autophagy takes place. But if you block this with the inhibitors, these functions are blocked. So even then, if you add camphorol, it cannot revert back. So that means in the absence of autophagic system, camphorol is unable to do the functions. That means it indicated that camphorol is really involving the autophagic processes or autophagic steps. So this was shown again by different proteins. I'll just skip this because of shortage of time. So I can just show you this slide. So you can see here, this is a BSA treated cell and palmitic acid LC32 level is very low. But if you treat the cells with camphorol, LC32 protein is very high. But if you add this wartmenin or the chloroquine, these are autophagy inhibitors, you can see the LC32 proteins are less. Even in the presence of camphorol, it cannot rescue to that extent as it was doing independently. So that means camphorol was using the autophagic steps for its function. Now, again, we showed that what was the pathway this camphorol was doing it. So MPK and mTORs are
Dr. Patra,
yes, my time is up.
yeah,
Yeah,
time
I
is
just
over. We have given an extra minute also since you had lost the time. But
so I just come, yeah, I come to the conclusion then. Yeah. So this is what we showed that mTOR is the molecule which is really inhibited by this camphorol, and AMPK is upregulated because that is how this is how it acts. And all these things we showed in the animals also. I'll just go back to my conclusion slides. Okay, so it's just I come to this one. So what will happen? We treat the animals with the high fat diets and this obesity, and if you treat them with camphorol, then the animals become obese, lean. So that means it was inhibiting the obesity condition in the animals as well. And this is same thing we could check for the glucose tolerance, fasting glucose, insulin tolerance, everything was taken care by this molecule. So again, you can see here plasma insulin, which was low in high fat diet, but with camphorol treatment, it reverts back. So all this together, and one important thing which I will show you, that pancreatic rejuvenation. So as you can see here, this is the controlled cells, high fat diet, all the pancreatic beta cells are gone. But if you treat them with camphorol, it rejuvenates. And when you do the immunohistochemistry of insulin, you can see here, this is the condition where it is highly improved as compared to the high fat diet stabilization condition. That means it was rejuvenating the pancreatic beta cells functions. So this is shown by different ways. So I'll come to the conclusion slides. So this is how it was acting. It was palmitic acid was inhibiting apoptosis, and that was killing the pancreatic beta cells, while this camphorol was inducing AMPK and inhibiting mTOR. That's how protecting the pancreatic beta cells function. So that is why it is a nutraceutical, can be used commercially for various applications. So that is how this is my lab who works for me, and this is my acknowledgements to the people who helped me in these works at different stages. So thank you very much. I can stop sharing.
Thank you. Thank you very much, Dr. Patra Roy, for a wonderful presentation. We have actually overshot the time, so
Yes.
we do not have any time for any questions. However, you can correspond with Dr. Patra directly through his contact displayed in the abstract book, and he will be happy, I am sure, to answer your question. Thank you once again.
Thank you. Thank you. Thank you.
Now this brings us to the fourth presentation, which is a gold medal oration. I request Professor Michael Avuldas and Professor A.V. Ramchandran to please come on to the stage. I will introduce the speaker. This is with reference to IS 23, Dr. Anurag Banerjee. I think, yeah. So over to Professor Michael Avuldas and Professor A.V. Ramchandran.
Good morning. This oration, named after and came back to India and joined as a faculty in the Pilani campus. He has about 28 research publications, which includes top 5 publications in biology of reproduction, general of molecular and cellular endocrinology, and general and comparative endocrinology. And he has also, in fact, in 2007, he joined the society as an annual member, and he also got the first prize for the best oral paper presentation. And after that, he moved out of India, and 2016 again he joined as a life member of the society. And he, in fact, he arranged the meeting in 2016 in Goa in a wonderful manner. And he has several ongoing research projects. And also he has been awarded gold medal orations from other societies in India and abroad. And if he could not come in person due to the pandemic situation, so I request the President, Dr. A.V. Ramachandran, to confer this fellowship on Dr. Arnav Banerjee.
Thank you, Dr. Michael. Good morning to all of you. So I shall read out the citation for Professor M.A. Akbar Shah and Professor Michael Aruldas, Gold Medal Oration 2020, awarded to Dr. Arnav Banerjee, Department of Biological Sciences, Birla Institute of Technology and Sciences, Pilani, R.K. Birla, Goa Campus, Goa, 4037. In recognition of his research contribution to the field of reproductive biology and comparative endocrinology by the Society for Reproductive Biology and Comparative Endocrinology on 28 December 2021, Mysuru, signed by Secretary Dr. Michael Aruldas and Vice President Dr. Suresh Venugopalan. Thank you.
Thank you very much, Professor A.V. Ramachandran and Professor Michael Aruldas for doing the needful. And congratulations on behalf of the chair and co-chair as well as the SRBC members present here to Dr. Arnav Banerjee. Dr. Arnav Banerjee is an assistant professor at Department of Biological Sciences with Pilani KK Birla Goa campus. He has already been introduced, and today he will be giving his gold medal oration on new insights in the grand old master hormone, mammalian gonadotropic releasing hormone GnRH1.
Thank you, Professor Devkar. Am I audible, please?
Yes, you're audible. It's over to you.
Okay,
And,
thank you.
We can see your presentation if you can just put it on the full screen mode.
Yeah.
And
So
you can put the curtain at the back side so that people can see you clearly.
okay, just give me 1 minute, please. Yeah, hopefully I came out from the hiding kind of a mode.
Now your face is looking like gold. For the gold medal.
Thank you. Maybe I'm matching my t-shirt also, but believe me, I'm feeling very, very dejected to be here. It's my, like I always had been, this is the family kind of an affair with me. SRBC, the first kind of a conference that I was associated when I was doing my master's in BHU, due to Professor Chandana Haldar, she had been so motivating, and I was always, into this kind of a conference. Particularly SRBC is like too close to me, so I cannot blame the condition what's happening around. I was so much excited to be there. So anyway, thank you to the organizers, to have given me a chance to share my work in this respected SRBC conference. And thank you for the organizers who take up. I mean, many of us have shifted from offline to online. So whatever we had requested, they had kept our request. And that's a huge kind of a deed. I had been a convener, so I know how much pressure it is. And they had been in this kind of a pandemic situation. Hats off to the organizers. Thank you to SRBC Executive Committee members and Professor M.A. Afghar and Professor M. Michael Arundas for have given me this kind of an opportunity to get this award in this from this prestigious society. The only thing that I couldn't be there, I just cannot, I mean, you can understand my kind of condition. So anyway, with this words, I would like to start my talk. So for all those who are listening, particularly the young ones, I like to tell them that how I started my journey in BITS. I joined BITS in 2013 after finishing my postdoc from abroad. And then when I came, I had to switch on to an entirely new field because my entire training was in embryo. But then coming to a private institute and to start with without an animal house to survive, it was like a huge challenge for me. But then a challenge makes people more perfect. I think, you have to come out of it and then you will find ways to come out. So something which you see my publication, you'll never see the word GnRH in any of them. But then I had taken up that field because something I expected, that I anticipated rather, that I could have done something in that field. So I started working with endocrinology, neuroendocrinology rather. And then because everything that I could think was from a cell-based model, not from an animal-based model. Then I took up obesity-associated cancer, some in silico prediction pathways, which had helped me to get a good number of publications. And then finally PCOS, which was very close to me. But then again, embryo, I have not yet started. So coming to the neuroendocrine part of whatever we had done, we know that fertility is one of the most important concern, no matter the fact that our country is the second most population-wise, it's the second most in number. But then within our country, and if you see in the world, there are two different trends that we could see. One of the trend is that we are over fertile, and the other trend equally that is visible is this, within our own country, there are regions which are subfertile, and presently where my institute is, Goa, there is a region where the fertility is tremendously low and the government is concerned about it. The same goes into the world scenario. So what I wanted to emphasize is that to work with fertility is important, and particularly when people, they make fun out of you and they say, oh, you are still working on fertility, when fertility itself is an issue in India. It's not that. We have to know more factors which can improve or manage fertility both ways. Now, all of us know that what this hypothalamus is. So it's hypo, it's below the thalamus, and that's why it's hypothalamus. But in function, it's nowhere below to anyone. It's the topmost in the endocrine system. So it controls all the other endocrine organs via release of many releasing factors. Now, this hypothalamus is a bunch of nuclei. Each nucleus is not the nucleus of a cell, but it's like a club of several cells which will release a common factor. And the different kind of cells that we see, the nucleus that we see in the hypothalamus are been grouped here. One of which is of my interest is the preoptic region. And it's in the preoptic region which releases GnRH and is also sexually dimorphic. We say that the sexual dimorphism exists between a man and a woman, and nowadays with orientational issue, this particular nucleus of the hypothalamus is of huge importance. Whatever the orientational alterations that we see, they are having a close correlation with any kind of a problem in this your preoptic region of the hypothalamus. But of course, it's also very, very important because it releases GnRH. Now, GnRH is a decapeptide. It has got 10 amino acids, particularly the amino acids 1, 2, 3, 6, and 10. These are the positions which are responsible for its structure as well as its function. The amino acids 5 and 6 and 7, that region, it's very susceptible for enzyme cleavage. So this particular decapeptide has got a very small half-life, and these amino acids 1, 2 and 3 and 5 and 6, they are responsible for the LH-FSH release. So what I'm trying to say, that though it's a very small kind of a peptide with 10 amino acids, it's very important. Each of these amino acids are very important to give its function and its structure. There are 3 different GnRH isoforms that have been present up through the vertebrate ladder: fish, then chicken Gnrh, and then the mammalian Gnrh1. I am presently working with only Gnrh1, that's the mammalian one, which we know that it is the it governs the endocrine axis. Now, what are the regulatory factors of Gnrh? Here, I would like to tell to all those young people who would like to come into this field, Gnrh is a wonderful molecule. Don't look at it just as a hormone. Look at it as a peptide which has been produced, and just like other peptides that have been produced from the cell, it has all these 4 levels of regulation. And because it's a secretory protein, it has the 4th one, that is secretion, which presently we are into. That is there a regulatory pathway that can be traced back. The other 3 today, if time permits, I'll show all the other 3 that we have done. Now, before we started, as I told, that I had never exposure to work with hypothalamus. So I took a little bit from what I had learned in my postdoc. We worked with embryos, and there what we found out is a new kind of a class of molecules that are known as polyamines. And these were instrumental in controlling embryonic development. A couple of papers, it showed that if there is inhibition of polyamine, there is loss of pregnancy, there is loss of implantation. So I picked up that polyamine thing and I started looking into literature, whether I could link it up with hypothalamus. Now, before coming to the state of art with polyamine, what are polyamines at all? Polyamines are biogenic amines. These are produced from the body from amino acid. I'll just show you. Now, when I looked into literature, whether polyamines have been explored in reproduction, I told you with embryo, a lot of work was done. In fact, with reproduction also, a lot of work has been done in relation to polyamine pituitary, polyamine gonads. What was missing was luckily this hypothalamic part, and that matched with my area of interest. So I just started excavating more. There was only one report when I started 2022, where it was been reported that polyamines might have some regulation with Gnrh in a neonatal rodent model. So that was something which was like a black box, which I started excavating. Now, what are polyamines? I told how polyamines synthesized. They're synthesized within the body. They're also obtained from dietary sources, but they can also be produced from arginine proline. And this is what is the biochemical pathway. The rate limiting enzyme is ornithine decarboxylase, or ODC1. And this causes ornithine to convert into putrescine, putrescine into spermidine, and then spermine. And this is kind of a circle which keeps on going. There is a strict balance that is to be maintained. And this is happening inside the body. Almost different parts of the body, they all produce polyamine to one or the other level. The words spermine and spermidine, of course, they have something to do with semen because they were first found in semen. That's why the name spermine and spermidine. And then the other one was putrescine. Now, so we started the work with polyamine and your hypothalamus and Gnrh. Now, when we started, as I told, there was only one report when 2002. Otherwise, there was no information left. However, they had done a lot of work with polyamine and different regions of the brain. So we wanted to first see how the polyamine expression they undergo in the hypothalamus. So we selected this model of the estrous cycle, which we all know, this rodent reproductive cycle, and this will give you a kind of an image how the polyamine expression alter in the hypothalamus. This is the pattern of the Gnrh that we show in the above panel, what we are showing. Then my student worked on categorizing the polyamine levels by checking immunohistochemistry of the different polyamines in the hypothalamus during estrous cycle. And we tried to correlate what is the correlation between the different polyamines and the different phases, and along with some markers such as Gnrh. And what we obtained was there was a strict positive correlation with polyamine and Gnrh. That was from the estrous cycle. Another student in our group started working on the aging model, because aging model is also an in vivo model been given where you can see the variation of polyamine very nicely in the hypothalamus. So we took young adult, mature adult, and old mice. Getting old mice was a bit tough because with the ethical clearance and issues, getting an old mice is not an easy way out. So we were lucky that we obtained it. Now, what we saw is Gnrh, as we all know, during the life cycle, during the adult phase, it will be increased. During the old, there is a significant decrease. We selected another marker which is known to the world so that we can use this as a frame of reference. And that was the Gnrh, that is opposite to Rh, Gnrh. And IH and RH, as they are opposite, their expression pattern in the hypothalamus are also opposite. As you could see these, I'm not showing the figures, I'm just showing the quantitative data just to save time. Then we went into the polyamine part again. ODC1, as I told, the rate limiting enzyme, spermidine, spermine, we showed we found out, as expected, that during old age, GnRH drops in the hypothalamus, the polyamine expressions also drop. The same pattern we obtained through qPCR study. So both protein and the transcript level shows the same. We again did the correlation and we found out similar thing, that GnRH as well as polyamine has a positive correlation. So what we could conclude from both the estrous cycle as well as the aging model, that these two, they are like in sync with each other, polyamine as well as GnRH. So the question is, do they really regulate GnRH? For that, we took the second part of the work. And the second part of the work, what we did, both in vitro and in vivo, we treated polyamine to the GT17 hypothalamic cell lines, which are known to produce GnRH, as also we treated the polyamine to adult rodents, females. Now, first we'll show you the in vitro data, and as expected, with polyamine, there is an increased expression of GnRH, and with its inhibitor, which is known as DFMO, there is a sharp decline, inhibitor of polyamine. This inhibits ODC1. And there is a sharp decline of GnRH in the in vitro. Same was obtained in vivo when we gave polyamine putrescine injection to the adult rodent mice. There's a significant increase of polyamine expression. Not only it could increase GnRH, of course, it could also lower GnRH. That's the first molecule that was been told to carry out both the thing. In one way, it's upregulating GnRH protein. The other way, it's decreasing GnRH. Following up, there is an upregulation of LH and FSH. Steroid synthesis also underwent upregulation. So this is something that it even functionally, it could produce more GnRH at the translation level. That is how the regulation was. Now we again impose a question. Can it only increase GnRH expression in a normal condition, or can it also alter GnRH expression under a stressed condition? And the stress that we wanted to give, we selected markers of aging because we were working on aging. Two hallmark of aging known to till date are lactate and TNF alpha. Lactate comes from the astrocyte to the hypothalamus, and this is an important metabolic fuel. But what happens is, in old age, there is lactate accumulation. Astrocyte keeps on supplying lactate, hypothalamus keeps on reserving it. That is inhibitory to its normal function. TNF alpha is another pro-inflammatory cytokine, which also keeps on accumulating during aging in the brain. And same is happening in the hypothalamus. It's well known that both lactate and TNF alpha decreases GnRH expression. And that is what we also obtained with giving lactate and TNF alpha. There was a decrease in GnRH expression in response to the control. What we did is we tried to put two different polyamines, putrescine and agmatine, in addition to lactate and TNF alpha. So what we found out very interestingly was, in presence of lactate and TNF alpha, putrescine as well as agmatine, the two polyamine, could rescue GnRH from its inhibitory action by lactate and TNF alpha. That was something which was very, very interesting, because it's known that in neurodegenerative diseases such as Parkinson's, Alzheimer's, fertility is compromised something, and there is high accumulation of lactate and TNF alpha in the brain of young people. In old, it's already reported, but in old, there's a downpour of GnRH. That also is known. But in a young people, if you have a high lactate, high TNF alpha due to neuroinflammation or due to some kind of neurodegenerative diseases, then you have to take care of fertility. That is something which putrescine, agmatine kind of molecules can come up with promising result, because it's showing that in the hypothalamus, it's capable of increasing GnRH even in presence of these stressful markers such as lactate and TNF alpha. Now, done all this, we impose a question: how on earth putrescine or agmatine is doing it? So we need to find out the mechanism. We did RNA-seq with our GT17 cell line. We wanted to find out what is the mechanism. We were screening through the data that we obtained through transcriptomic analysis of the GT17 cells after treatment with putrescine. We were not getting the classical pathways getting altered, but what we got very interestingly was the biological clock rhythm. Those pathway genes are getting altered due to putrescine treatment. Now we wanted to correlate how putrescine via biological clock rhythm pathway can correlate can upregulate GnRH. That was the ultimate goal, that how putrescine can upregulate GnRH and can rescue it even after treatment with TNF alpha. So what we did, we validated the data. Now, due to time constraint, I cannot explain the entire pathway for biological clock rhythm. in cut short, it is reported that GT17 cells, they expresses the major markers such as period 1, period 2, period 3, cry1, cry2, then clock genes in the GT17 cells. So GT17 cells, they can undergo rhythmic expression of these clock genes. With expression of putrescine, I mean, with treatment of putrescine, what we saw was these clock genes were also getting upregulated, which in the downstream upregulated GnRH. If you could see, there is we carried out a 6-hourly time-dependent experiment. We treated the GT17 cells with putrescine, and after every 6 hours, we collected the RNA. We then checked the expression of the clock genes, that is your biological clock genes, that is per2, per1, per2, per3, cry, and then clock BML1. I'm not showing the entire data. It almost matched with the pattern of GnRH that we obtained. That is, they are being controlling the GnRH. And then we look back into literature. Is there a correlation between these clock genes and GnRH? There is a report which was shown clearly that clock genes regulate GnRH expression in GT177 cells. What was not known, upstream to the clock gene was putrescine, which could go and modulate GnRH expression through this. Quickly coming down to the next part, that is transcriptional regulation. Yeah, somebody wanted to say something? Okay, fine. So transcriptional regulation. In the transcriptional regulation, as I told you, kindly look GnRH as not a hormone. Look at it as a protein. Now, we got an invitation at that time to write a review from the journal. So we wrote and we drew this figure. After doing this work, we were more interested to excavate that for the transcription of this GnRH gene, there are some well-known specific transcription factors, and these transcription factors could be clubbed into inhibitory, excitatory. Till date, there was no information available. During aging, GnRH lowers, the world knows, but whether these transcriptional factors also undergo some kind of alteration was not known. What interestingly we found out, all the repressors, they were high during old age. All the repressors and all the activators, they were high in the adult age. Some of the activators you also see high in the old age, because those transcription factors, they are not so specific for GnRH. Some of them are also doing some other activities which gets high in senescent period. So that's why you see that high kind of expression. Then what we did, we again treated the cells with TNF-alpha and lactate. These are the two hallmarks of aging to the GT17 cell. Now, the earlier one was the in vivo data. What we wanted to see, whether that could create an aging-like condition. We looked at some of the well-known anti-aging, oxidative stress markers, and we saw that with lactate TNF-alpha, there was upregulation of the oxidative stress markers as well as downregulation of antioxidant enzymes, proving that there was some kind of that condition being created by treatment of lactate and TNF-alpha. We checked the GnRH transcript level. It was also been decreased. Now we went to check all the transcription factors. As expected, we saw similar kind of pattern. I'm not explaining in detail. We saw even the expression of NF-kappa beta that was also upregulated due to lactate and TNF-alpha. At the same time, we went to, we fished out the transcriptomic data of a single hypothalamic cell, and we wanted to match whatever we obtained. Does it match with that? We were very happy to know that similar kind of analysis we did, and we got similar kind of molecules that from the transcriptomic data of a single hypothalamic cell during aging in comparison to young, it shows upregulation of lactate that goes and downstream and upregulates TNF-alpha, which upregulates molecules such as NF-kappa beta, and then whatever transcription factors that we see were also getting upregulated, as a result of which these repressors downregulated your GnRH in an adult state. So old neuron, old mice, what happens, old brain rather, I should say, even in us, there is high supply of lactate coming from the astrocyte, and this astrocyte supplies this lactate to the neuron. In an adult stage, the neuron utilizes the lactate and carries out other metabolic function. In an old age, it cannot. It rather accumulates lactate. This lactate accumulation causes upregulation of the pro-inflammatory factors, the transcription factors such as NF-kappa beta and other repressors of GnRH goes high, and as a result of which you see a downregulation of GnRH during old age. So this is the transcriptional regulation part. Now coming to the last part of my work for today, what I want to show is the post-transcriptional regulation of GnRH. GnRH, as I told you, is a wonderful molecule in the fact that it shows all the levels of regulation, one of which is also alternate splicing. We all know what it is, that from a single gene we get different different transcript because of alternative splicing. Now GnRH has got 4 exons as it is shown, and these 4 exons, they are in the functional GnRH will get all the 4 exons retained as it's expected. But there are other 2 variants that are also being shown, which either undergoes an intron A retention or undergoes exon skipping. So in both cases, they would not produce GnRH because the functional GnRH molecule is not been reflected in its mRNA, because either it is losing one of the exon or it's retaining one of the introns. So these are the 3 the GnRH alternate splice variants. One is the functional E1, E2, E3, E4. The other 2, one doesn't have E2, that is V2 variant, and the V3 variant, it includes an intron. Intron is not to be included.
It's already 5 minutes.
Yeah, I'm just finishing it up.
Please continue.
I'm just finishing it up, Professor Devkar. So we selected very specific primers and we did a sequence analysis of V1, V2, V3, because if we had done a wrong primer analysis, we might get wrong expression. It almost matched about 90% with the whatever length of the products that we got. It matched with 90% with whatever was been submitted in NCBI. So we were very much confident that the primer designing was correct. And then we checked the expression of V2, V3. V1, everybody knows. V2, V3, almost, 5% of information is known. That of course V2, V3 exists, but what is their expression, variation during aging, during metabolic stress that we created was not known. We checked specific splicing factor expression, which is very specific for GnRH splicing. We obtained a variation there also. Then what was we questioning is, if there was V2, V3 that is undergoing variation just like V1, then are they getting translated? The answer we obtained till date, the information is just known that V2 and V3, they are not translated. But if they are not translated, what are the reasons behind it? Nobody has given that information. We are going to communicate this work very soon. Through in silico prediction, what we found out for V3 variant, it cannot, the V2 variant cannot be translated because it has a stop codon in between. V3, it has no premature stop codon, but what it has, it has in its sequence some of the translation inhibitors to bind to, such as PUM2, SAMD4A, and some of the microRNAs. Their binding sites are located within the variant 3, which decreases their translational efficiency. And as a result of which, we don't get V2 and V3 into product, but V1 is getting translated as GnRH. But now our question is, if V2, V3 is not getting translated, why they are expression getting varying? So what is the function of it? We are still into that work. We are trying to see through informatics what V2, V3 as RNA can do to regulate maybe some other genes, or might be equally important for regulation of GnRH. That's a very important black box that we have hit upon. We don't know how far we would be going. So this is our team situated in Goa. We quite a number of time goes out. We go out for venting out of frustrations. Not only students get frustrations, even as a PI, sometimes I too get frustration. But the best way to vent out is to go and have a dinner all together, and then things get solved very fast. I am thankful to all my collaborators at BITS Goa, NIRH Mumbai. We'll soon hear Dr. Shravani Mukherjee. So I'm thankful to her for giving me some of the animal work that I have not shown today, but we did it there. I'm thankful to Department of Zoology, Banaras Hindu University, Varanasi, and I'm thankful for we'll soon start some of the human work also with collaboration of NIRH Mumbai and with Sinai IVF Clinic Hospital Goa. And I thank my funding agency, SCRB and DBT, and thank you to all. I'm here for any questions.
Thank you, Dr. Arnab Banerjee, for taking us all through a wonderful journey of the GnRH and your experiences that you narrated. It was very nice and interesting. Since it is a gold medal ovation, we will not have any question answers for it. However, if you yet have the questions, you can interact with the Dr. Arnab in person directly. I once again congratulate you, Dr. Arnab Banerjee, for the gold medal ovation, and hopefully next to SRBC you are there in person. as a part of the society. Thank you.
Thank you to all of you all. And please feel free to write if you have any suggestions or any questions. Thank you.
Thank you once again. With this, we come to the last presentation of today's morning session. IS 24 is the invited lecture, invited talk by Dr. Srijit Panikar. He is an assistant professor at the Department of Zoology and the Center, Advanced Center for Regenerative Medicine and Stem Cells in Cutaneous Research in the University of Kerala. Dr. Srijit Panikar is going to talk on hair loss induces an defective fatty acid metabolism and mitochondrial dysfunction. Dr. Srijit, your voice is muted. If you can unmute yourself.
Am I audible?
Yes, you are audible now. You can put your slides on the presentation mode, and you have 15 minutes time with 5 minutes for interaction. Over to you.
Good morning. First of all, I thank the organizers, Professor Malini, to give me this opportunity. And I also thank all the members of the SRBC to present my work in this prestigious conference. And I'm basically working on hair and hair loss and their regenerative potential. And today I'm specifically talking about how hair loss is induced by defective fatty acid metabolism and mitochondrial dysfunction. And we know that hair plays an important role in people overall physical appearance, self-perception, and also act as a protective barrier. And it has a lot of functionality, and it has an important role in thermoregulation, sensory perception, species recognition, and neuroreception.
Put on the slideshow.
Hello?
Please put on the slideshow.
Yeah, sorry, it is in slideshow right now.
Press F5 so that it is in the presentation mode.
Yeah, it is in presentation mode now.
It is not. We can just see the slides. Stop the sharing and reshare it so that it will be done properly.
Now it is fine. Hello?
Yeah, it is not yet seen, but then you can proceed. You can change the slides as per your thought. I suggest that you share your screen so that whatever is there on your screen is seen. Full screen.
Hello?
Yes, you may continue.
Oh, there are
Yes, you may continue. You may
Hello?
continue.
There is an essential role. The hair follicle have their own immune prevalence mechanism is there, and if any changes in the immune prevalence mechanism, specifically in the hair follicle, that adversity affect the hair health, and that condition we call it alopecia. There are different factors are actually contributing this hair loss condition, and like aging, genetics, some medication, malnutrition, and autoimmune disorder, and chronic inflammation, and these are the conditions actually activate hair loss. In addition to that, there are other factors also contributing hair loss conditions, and our lab is specifically working on these factors like inhibition of cholesterol biosynthesis, how the cholesterol biosynthesis inhibit hair loss, and Then another important condition we are looking, alteration of fatty acid metabolism, and how the fatty acid metabolism affect the hair health, and any changes in the fatty acid metabolism, how the immune function is actually disturbed, and that initiate the permanent destruction of hair follicle. And in addition to that, we are looking how the nuclear receptors like pepper gamma have any role in this particular condition. And then after that, we are looking the environmental pollutants like dioxin is regulating the hair loss condition, or it is affecting the hair loss or not. And these are some of the major areas we are our lab is currently working on. And all these important factors are very much interlinked. And to understand the class, before moving to the study part, I'm just briefly introducing the alopecia. There are two important type of alopecias are there, that is calling scarring alopecia and non-scarring alopecia. And this is the important classification of alopecia. Non-scarring alopecia is actually affecting the in the bulb region of hair follicle. And this is the area basically affected in the scarring alopecia, and there is no scar formation in this particular condition. And two important types are basically these are the examples of non-scarring alopecia: androgenetic alopecia, telogen effluvium alopecia areata are the best example we can see. And the scarring alopecia is again divided into different categories, depends upon the type of inflammatory cells in the sebaceous unit. Hello.
We can't see your slide changing. We can just see the slide number 2. That's it. Are you yet talking about the slide number 2, or you have changed this? Yeah, now it has changed. Yes.
Okay. And this is the basically the alopecia is classified into two different categories: scarring alopecia and non-scarring alopecia. Scarring alopecia is divided into basically affecting in the bulb region of hair follicle where the hair follicle, where the stems, where the derma papilla cells are present. And the androgenetic, they are androgenetic alopecia and alopecia areata. And the scarring alopecia is again divided into three different categories, depends upon the inflammatory cells present, like lymphocytic, neutrophilic, and mixed. And the lymphocytic are lichen planopilaris, frontal fibrosing alopecia, central centrifugal cicatric alopecia, etc. And the frontal fibrosing alopecia, the major type of alopecia we are actually currently working on. And because this prevalence of this particular condition is very much increasing, and neutrophilic, where the neutrophils are predominantly present, mixed means mixed type of like neutrophils and lymphocyte are both are present in this condition, and there will be a severe inflammation in this particular hair loss condition. To understand this condition, hello.
Yes, tell me.
Presentation is visible right now, or?
Yeah, you can go. You can talk. We can see the slide change. Please go ahead. We are able to see slide number 4 right now. Crosstalk relation establishing.
Yeah, to understand this pathogenesis condition, and we are thinking that some type of lipid metabolic changes are happening in this particular condition. To understand that, we did gene profiling of around 40 patient samples, and the studies we identified that there will be a decreased lipid metabolic genes are altered, and their expression level is very much down regulated. And here we can clearly see that the green represent the down regulated genes compared to the normal sample. The unaffected and affected samples, the gene expression of lipid metabolic genes are significantly affected, and that's showing that the lipid metabolism is happening in the early stage of the disease. And similarly, we also identify that there will be an activation of xenobiotic responsive genes like CYP genes are very much activated activated. This showing that there will be a activation of xenobiotic genes, and there will be a down regulation of lipid metabolic genes, and that showing that there is an interaction between these two pathways present in the hair follicle or alopecia pathogenesis. And we also identified that there will be a interaction between nuclear receptor and hydrocarbon receptor signaling pathway, and the studies clearly showing that activation of pepper gamma, inactivation of pepper gamma by the AHR signaling, and there will be an activation of pepper gamma due to the aryl hydrocarbon receptor after the treatment of AHR, and there will be an inactivation of AHR by the activation of pioglitazone, and there is also showing that like clean crosstalk regulation is happening. And to understand more that we identified that there is a lipid metabolic genes are activating. Similarly, there will be an activation of these pepper gamma. And to understand more about this thing, and we are looking the metabolic analysis of these patient sample, and what are the key biochemical pathways activated after this pathogenesis condition, and we are looking in the metabolic profiling of the patient sample, and one of the animal model we are actually have very much similarity like alopecia sample, and we are selected an animal model, and that animal model aryl hydrocarbon receptor is constitutively activated in this animal model. And in the picture we can clearly see that AHR, while the wild type individual, there is no hair loss, but in the AHR activated model system, there is a hair loss happening in the early stage of the condition, like 3 weeks itself, there will be a progressive hair loss is happening. And that's showing that there will be a lot of similarity in the case of alopecia patient sample and the transgenic model. And this is the histological analysis of the wild type individual and AHR individual animal model. And the animal model, there is a progressive hair loss is happening, and the histology itself it is clearly showing. And the 3 week within 3 weeks the complete hair loss is happening. And in addition to that, there is another changes also happening in this particular animal model, like there will be a less sebaceous subcutaneous fat, inflammatory response are very much high, lymphocyte institutes are predominantly present in this particular condition. And another important condition is that follicular plaque.
What number of what is the slide number you are talking about right now? We can just see the fourth slide. I assume that there is some network issue in your. Yes, now it's better. Now it is better.
Yeah, I'm sorry about that. I don't know what is happening. Really sorry for that. Now it's is it visible?
Yes, we can now see the slide with the histology photograph.
Okay. To understand the PCA pathogenesis, we are actually identified a model system, and this model system is the mice model we are actually taken is a transgenic model, and this model aryl hydrocarbon receptor is constantly activated, and this this is the picture showing the wild type individual is highly there is this is a wild type individual, and the AHR model system there is no hair loss, and we identify that there is a developed progressive alopecia in the 3 week onwards. That showing that this model system we can use as a study model to look alopecia pathogenesis. And we also did the profile gene expression profiling, histological analysis in different stages, like 0 week to 9 week, compared to the, and we compared with wild type individual to AHR individual. And we can clearly see that AHR individual, there is a complete hair loss in the 3 weeks, and the histology structure histology itself showing that there is no hair loss, and there will be showing some accumulation of some metabolites in this particular stage, and there will be a severe inflammation, and follicular plugging are the hallmark of these particular stages. And that's why we use these model systems to study the alopecia sample. And this is the next slide. Is visible?
Yes, it is visible.
Okay, then we are looking at the comparative analysis of this patient sample, like frontal fibrosing alopecia and AHR mice model. And we can, our aim is that there is any relation between the this metabolite activation in this particular patient and the AHR model system. And that's why we are actually comparing the metabolomic analysis in AHR model and the fibrosing FFA patient samples. And this is the expression studies of these both samples. And my second, the other slide is visible. Next slide is visible. And this is the gene expression profile of unaffected FFA and FFA samples. And compared with, we are looking the 1 week AHR mice. And affected FFA, the lipid metabolism are the top pathways. And similarly, we can also see in the mice model also showing similar pathways are affecting. That showing that there is a lot of similarity between this model system and the patient samples. And we also looked the signaling pathway, and we interestingly we identified that lipid metabolic genes are dysfunction is the predominant pathway in affected sample, and similarly the oxidative stress phosphorylation also affected. And then 1 week mice model also showing similar character features, xenobiotic metabolism, fatty acid metabolism, all these are actually disturbed in this particular condition. That's showing that this both condition are very much similar. And to look more about this process, and we just look metabolomic analysis of these two patient samples, the patient sample and mice model. And the metabolites, we know that this is very much affecting the phenotypic nature of the individual, and we think that these metabolites probably have regulating the inflammatory condition in this stage, and that eventually induce the hair loss condition. And this is a statistical analysis we did. We are comparing with FFA with unaffected, FFA unaffected with normal, and affected with normal. Similarly, each stages we are actually each stages we are actually looking the analysis, like 1 week, 2 weeks, and comparatively wild type individuals also we are analyzed. And this is the defective. This is the metabolites we are actually identified in the fat that probably very much related to mitochondrial metabolism and fatty acid accumulation. And compared to normal, we identify that the metabolites like carpolate, deconate, and pelgranate are very much activated compared to the normal sample. And this showing that this activation of these metabolite probably attract the inflammatory cells to the hair follicle and induce the hair loss condition. And similarly, we also identify that there is a severe fatty acid accumulation in long chain and medium chain fatty acids are accumulated in the mice model also, like long chain fatty acid like henicosinate and behenate are the metabolites are actually activated, and this activation is very much higher in the 3 weeks of 3 weeks individual, and this is a 3 week individual. And 3 week individual, we can see that compared to the normal, the wild type individual, the AHR individual have high level of metabolites, and that's showing that these metabolites are very much accumulated, and that have affecting the health of the hair follicle. And in addition to that, we looked at mitochondrial metabolism in this particular condition, like TCA cycle we are actually checked, and we identified that the unaffected and affected sample compared to the normal, the pyruvate level is very much inactivated, similar like citrate, fumarate, all these metabolites are severely affected. That's showing that metabolism, the mitochondrial metabolism is disturbed due to this particular condition, and that actually adversely affect the health of the hair follicle.
The time is over. If you can wind up.
And in addition to that, we also check the glutathione reduced and the glutathione oxidate, and you can clearly see that glutathione oxidase is very much activated. That's showing that there will be a oxidative stress condition is activated in this mice model and the patient sample. That's showing that the mitochondria health, mitochondria will be affected, and that actually activating the hair loss condition, or adversely affect the health of the hair follicle. And next, we are actually looking how these metabolites are actually affecting the hair loss condition. And we check the in vitro analysis. And in vitro, we used primaries like human hair follicle outroot sheath cells. And then we dose-dependent manner, we added these metabolites already activated in the patient sample and the mice model, and clearly showing that the inflammatory genes like interferon, TLR, TLR4, TGA beta, NF kappa B, MMP, everything is activated. And there will be a pelgonic also showing. Hello?
Yeah, your time is over. If you can.
Yeah, I'm finishing that.
Your presentation, please.
Yeah. And similarly, the pelgonic acid also showing the activation of inflammatory cells and the downregulation of genes like stem cell genes like LGR5 is actually downregulating. This clearly showing that there will be an accumulation of fatty acid metabolism or disruption of that actually induce inflammation and reduce the stem cell function in the hair follicle, and that adversely affect the health of the hair. And we also check the TEM analysis, and we can clearly see that in the AHR mice model, the mitochondrial structure is drastically affected, and this is the normal mice, normal mitochondria, and there will be a mitochondria is severely affected with this condition. And we also check the mitochondrial functionality, mitochondrial morphology in patient samples also, and this is the normal individual. And this is unaffected individual. There is already some changes happening in the mitochondria, and there is a severe destruction is the mitochondria in the affected individual, and that's showing that the accumulation of metabolites in the hair follicle actually adversely affect that create mitochondrial dysfunction and eventually activate the hair loss conditions. And this is the working model. We can there will be some environmental factors, genetic factors, and metabolic disorder activate aryl heterocarbon receptor, and that actually inhibit pepper gamma and adversely affect mitochondrial dysfunction due to the defective mitochondrial beta oxidation, decreased energy metabolism, and accumulation of fatty acid, and that eventually leads to the ROS generation and oxidative stress. And this oxidative stress condition induce inflammation, stem cell host, and eventually that adversely affected the follicles. And that is the way the alopecia FFA is predominantly activated in this. The prevalence is going up because these metabolites are actually accumulated, and that actually inducing the hair loss conditions. Thank you. And I acknowledge KCST, Department of Health, ICMR, and Government of Kerala.
Thank you, Dr. Srijit Panikkar, for your wonderful presentation. Since Dr. Srijit has already overshot the time, we will not have any questions. However, if you have any doubts, queries, or suggestions, you can talk with him, you can correspond with him directly. With this, we come to the end of this morning session. I request my co-chair, Professor Dr. Sivanandappa, to sum up the session. Sir.
Thank you. Today's session somewhat of a great interest
from geographical point of view as well as from the science point of view. We started in New Zealand, went over to United States, came back to India in Uttarakhand, then went to the coast of Goa, then ended up with Trivandrum coast. Professor Cochrane in New Zealand gave an interesting talk how the stress causes the change as indicated by particular on the birds, human handling, and he showed examples for two kinds of personality that could possibly exist that could handle stress from fish to birds. This was followed by Dr. Gnrh in the United States, who gave a talk on transplantation biology, which was followed by the gold medal audition by Dr. Patra Roy. It was followed by a lecture by Dr. Patra Roy. He spoke about a well-known compound that's available for researchers to show that it could protect against cellular injury and cell death that causes diabetes. And he showed how the mechanism that could be involved in protection. This was followed by the gold medal oration talk by Dr. Aruna Banerjee in Goa, who showed us that the master hormone Gnrh is not a great master of form because it is also controlled by minor players. How minor they are, they showed us starting from polyamine to TMR. So therefore, Gnrh hormone regulation is subject to control by the mechanisms that could be happening in the brain, the hypothalamus. And the last talk from our Srijit Panikar from Trivandrum, who started quite familiar to us for many years about alopecia, hair loss, and the mechanisms involved. And I hope next year again he will tell us more about this on hair loss mechanisms using the newer data that's probably being generated in that. So with this, we end this session of online talks, inverted talks, and we thank all the speakers, both abroad and from India, for giving us this opportunity to present their scientific findings at this meeting. Thank you, and thank you, Dr. Joseph, for conducting the show efficiently. And luckily, we didn't have a major explosion. And thank you for all your patience. Thank you.
Thank you to all the speakers. As a token of our gratitude to the chairs, I request Dr. G. Subramania to award mementos to them.
Please.
Thank you, sir. We shall now break for tea. Since the session ended quite late, we'll start the session time at 11:15. And I request Jyoti Chaitanya to give her presentation for the session 11. Sorry, give her PowerPoint presentation.